Hi, I'm John Hamill, and I'm the Flight Operations Manager for the Student Spaceflight Experiments Program. In the following videos, you'll meet teams from the US and Canada that are participating in SSEP Mission 13 to the ISS. They'll discuss their experiments and what they hope to learn. I hope you have a great day and enjoy the launch.
Hi, we're the Mung Bean Team 2018 from Hillsborough County, Tampa, Florida.
Our team sent mung beans up to the International Space Station to analyze the germination rate between mung beans in microgravity and mung beans on the Earth's surface.
Our team decided to use mung beans whenever we researched plant types and found out that they had a lot of nutrients and proteins that should be beneficial to astronauts in space.
From this, we hope to learn the germination rate between mung beans in microgravity and on Earth's surface. We also hope to learn more about growth of plants in microgravity.
We are Career Magnet Academy from the Student Spaceflight Experiment Program in Knox County. We are researching how to produce a synthetic soil from waste generated on board the ISS. During our first meeting, we were interested in how to sustain life on other planets. In order to sustain life, a source of food is necessary. Lack of soil is a problem. Microgravity may also turn out to be a problem. In our experiment, we hope to recycle waste using mycelium spawn to produce fertile soil that will grow microgreens. For example, this is a large-scale product that we did in mason jars. This first jar is an example of our waste, and in the second jar you can see the mycelium starting to decompose our waste as it grows through it. And in the third jar you can see the outcome, and you can see that the mycelium produced mushrooms and a composted soil. In the future, astronauts will be able to recycle their waste into soil, producing a variety of foods.
Alright, I'm ready. We are space pirates from the Student Spaceflight Experiments Program at Berea Community High School in Berea, Kentucky.
Our experiment is rate of rust formation in microgravity.
We will be testing how quickly rust forms in microgravity and on Earth, and seeing which will rust more quickly.
We believe that the rate of rust formation will be more in microgravity.
After coming up with the idea, we chose this topic because we figured it would be able to be in the parameters set, those type parameters set by the SSEP, and we were also curious on how rust formed in the first place, so that's why we chose the topic.
We are Team Quinoa from the Student Spaceflight Experiment Program in Clark County, Nevada. Our experiment addresses the question, will quinoa grow in microgravity?
We chose quinoa because it is very high in protein and nutrients. Quinoa is also very high in fiber, phosphorus, magnesium, iron, potassium, and calcium, B vitamins, vitamin E, and many other beneficial antioxidants. It is also gluten-free. For experiment, we used spring water, rock wool, fertilizer, 10% neutral buffered formaldehyde, and quinoa.
The reason why quinoa interests us is because it's been used in South America for thousands of years as a food source.
Our research told us that 70% of the quinoa seeds will germinate and grow.
We want to learn not only if quinoa will grow in microgravity, but also if the seeds will germinate and grow at the same 70% rate as on Earth.
One thing that we've already learned is that we could have added enough fluid to double the amount of fluid that we originally had in the mini lab.
This project has been a lot of work, and we are excited that our experiment was chosen for Mission 13. Thank you.
We are the Yeasty Boys from the Student Spaceflight Experiments Program at Stockton University in Galloway, New Jersey. And the title of our experiment is Analysis of Double-Stranded DNA Breaker Pair and Haploid Saccharomyces cerevisiae under spaceflight conditions.
As we are spending more time in space, there's an increased amount of radiation and DNA damage. Our experiment aims to see if DNA can be repaired under these conditions.
So basically, the point of our experiment is to send bug and yeast into space and treat them with bleomycin, which is a DNA damaging agent. After this treatment, we will immobilize the yeast with paraformaldehyde and send them back to Earth for analysis.
So the main reason why we chose this topic is because it intersects genetics and medicine, which are two fields that we're extremely interested in. And what we're hoping to gain out of this is learning more about DNA damage repair in space so we can improve astronaut health. And space is cool.
Easy Boys out!
We are Logan Ford and Christian James from the Student Spaceflight Experiments program in Hershey, PA from the Milton Hershey School.
Our project is the effect that microgravity has on algal cyst lipid production. This project is meant to check if microgravity is a stress factor, and the reason why is because when algae is stressed, it produces more lipids, but with it, they grow slower. Algae lipids are the fat cells that we break down to release the oil and make the biofuel.
The reason why we chose this project because the future is biofuel, and if we can't find a more efficient way to grow algae lipids, then the entire process of making biofuel is a lot harder. I was learning about algae in my biology class, and I wanted to put use into my learning.
I've always been interested about biofuel because it could actually change the world, or at least slow down our pending doom. Because if we grow algae for biofuel, then we can make the atmosphere carbon neutral.
We hope to learn if we could make life better with biofuel, if microgravity is a stress factor, to algae making more lipids equaling more biofuel.
Hello, we are Hunter and Eliza from the Student Spaceflight Experiment program in Hershey, PA.
The title of our experiment is The Effect of Microgravity on the Adhesion and Curing of Oil-Based Artist Paint. Our experiment aims to test how oil paint adheres and cures in space compared to here on Earth.
We both have a love and appreciation for art and space. Our motivation for this project is to improve the mental health of astronauts while they are up in space by giving them a creative outlet.
We hope to learn that oil-based paint is a viable medium for astronauts to use up in space.
We are Trey and Jacob from the Student Spaceflight Experiments program in Hunter, North Dakota. The title of our experiment is Key for Growth.
How did you choose this investigation?
We chose this investigation by searching healthy foods up on the internet. One of the first options that came up was kefir grains. We chose them because they are healthy and they are reusable.
What interested you about this topic?
We were interested by kefir grains because they build lean muscle mass and help your digestive system. They're also reusable and healthy.
Tell the description of the experiment.
In volume 1, we have 8 milliliters of distilled water. In volume 2, we have 4 kefir grains and 0.9 grams of granulated sugar. The astronaut will remove the clamp and shake vigorously for 45 seconds to combine them together.
What do you hope to learn?
We hope to learn about how microgravity affects the growth of kefir grains in water.
We call ourselves the Tomato Group. We are students from all over Wisconsin and attend iForward Online Charter School, located in Grantsburg, Wisconsin. We are honored to be presenting our experiment for the Student Spaceflight Experiment program. Solanum lycopersicum, or red velvet cherry tomatoes, are extremely nutritious and easy to grow. We will have 6 seeds and vermiculite in the middle portion of the FME tube. Once in space, the astronauts will release the water from one side of the FME tube, and before returning to Earth, the fixative will be released, which will preserve the germinated seeds. Despite being in different physical locations, we had a wonderful time experimenting together in a virtual classroom. We were interested in growing this variety of cherry tomato in microgravity because it is a species that has been grown on the ISS before a shorter duration. The seeds are tiny enough that 6 will fit very well in the FME tube. This species of tomato is also extremely nutritious. We hope to further past findings and show that the cherry tomato could be a successful and sustainable crop and provide amazing nutrition in space.
We are the Rust Investigation Team from the Student Spaceflight Experiments Program at Wiley Intermediate School in Watertown, New York.
Our proposal title is Rust Investigation. In this project, we are seeing if we are able to remove rust faster or slower in microgravity.
Our original idea was to see if we could rust iron in space, but there's no oxygen in space, so we all decided on removing rust instead.
This project interested us because in the future, if mankind has to live in space, they'll still need oxygen, which can cause important items to rust. If an important piece of machinery rusts, everyone can be in danger. But if we know how to remove rust in microgravity, we can work our way towards a safer future.
We hope to learn if microgravity affects how fast rust is removed from metal.
We are the Cosmic Comets from the Student Space Flight Experiments Program in Kansas City, Kansas.
The title of our experiment is the growth of mint in microgravity. We want to see if mint can grow in microgravity, because if so, it can be beneficial to astronauts.
We chose this investigation because we wanted to help astronauts. We know that mint has a lot of health benefits, such as reducing fevers, soothing sore throats, and helping upset stomachs.
If our experiment is successful, it will prove that we can grow mint in space. Not only will it prove that we can grow mint, it will also prove that we can grow plants in general. If we are capable of growing plants in general, we could explore space without constant need to resupply.
Hi, I'm Maxwell Brown from the Micro-G team for the Student Spaceflight Experiment Program in Klein ISD. Our project this year was based on protein crystallization. Now, previous research has demonstrated that adding a monolayer, which is like a little film on the surface of a container, can improve the efficiency of protein crystallization, as well as microgravity can do the same. So our research is attempting to bring both of those effects together and to see whether we can get some sort of greater efficiency of protein crystallization than either one of the two effects would on their own. So as such, our project is called The Effect of Self-Assembled Monolayers on Microgravity Protein Crystallization, and so that's what we've been working on. Where we actually originally got this idea was one of the members of our team, his name's Michael G, and last summer he went and worked with the research team run by Dr. Randall Lee at the University of Houston, and their research focused on these self-assembled monolayers. When we heard about the Student Spaceflight Experiment Program, we wanted to try and apply this knowledge to the protein crystallization and things like that. From this experience, we basically just want to get a feel for how scientific studies and experiments are done in the real world. All three of us, a lot of our experience with science has just been in the classroom, and through SSEP, we've had the opportunity to get a hands-on experience with the research and to get a feel for how research is actually conducted in the real world. All in all, just thanks for this great experience. I speak for all three of us when I say that our lives were really changed by the opportunity to participate in the Student Spaceflight Experiment Program.
Hello, my name is Colette Folks, and I'm part of the 6-member Marfa Martian team from Student Spaceflight Experiments in Marfa, Texas. Today we are sharing our experiment, Eradicating Bacteria in Microgravity. We chose this experiment because a harmful strain of bacteria, Bacillus cereus, was found on the space station, and it may endanger the astronauts' health. Now each of the team will discuss a part of the experiment.
I'm Madison Cash, and we're interested in this topic because we wanted to study something that could help the astronauts. Because Bacillus cereus is harmful, we used a safer but related bacteria in our experiment, Bacillus subtilis.
Hi, my name is Ashley Surian, and I'm a co-principal investigator of the Marfa Martians team. The bacteria Bacillus subtilis is commonly found in the environment and in our intestines. This bacteria grows spores to protect itself from any harm or danger. We plan on using isopropanol to eradicate the bacteria before it has time to germinate any sort of spore.
I'm Daniela Fernandez, and we're here to learn that an antibacterial agent, isopropanol, will eradicate Bacillus subtilis with greater effectiveness in microgravity than it would in Earth's gravity. To do the experiment, we use a Type 3 FME mini lab, which is approximately the size of a straw. Charlotte will explain further.
Hello, I'm Sean Browning, and I will be explaining our experiment by using this pool noodle. In the first compartment, we will have our bacteria, freeze-dried bacteria. In our second compartment, we will have the media broth, which will wake up the bacteria. In our third compartment, we have the isopropanol. First, we will release the clamp between compartments 1 and 2 and shake for 15 seconds. And then we will unclamp clamp 2 to shake them all together for 15 seconds. And hopefully the isopropanol will kill the bacteria.
Hi, I'm Mabel Melgard. Following the experiment done in space as well as on Earth, samples will be stained and placed on slides, and we will view them under a microscope to get a bacteria count. We will learn how effective the isopropanol was on the bacteria in microgravity and compared to Earth's gravity. Thank you, SSEP!
Hello, we're conducting the Student Spaceflight Experiments Program, Mission 13 from FAR, Texas, and our experiment name is What are the effects on microgravity for the cellular growth of Spinia ulceosa?
How did we choose this investigation? The reason that we chose this investigation is because we want to know how vegetation is gonna grow in space. And the reason for the spinach is because it usually grows within the month that it is going to be conducted on.
What did we hope to learn? Well, I hope to learn on how growing spinach in space can be useful, such as how the effects of microgravity can weigh in this particular situation, such as how complex growing a plant can be in space and how vital it would be for the astronauts.
What interested us about this topic? Well, space has always been a mystery, and it is part of the future, and we need to be part of it.
Okay, so we had a great experience working with the SSEP program, and we're looking forward to the end result for the effects of microgravity on cellular growth for Spinia ulceosa.
Hi, I'm Dave Katz.
I'm Mo Kashkush.
I'm Anu Patel.
We are the University of Pittsburgh School of Pharmacy with the Student Spaceflight Experiments Program in Pittsburgh, Pennsylvania. Our experiment is the transcriptomic analysis of Escherichia coli response to ciprofloxacin and microgravity.
We're sending an antibiotic and bacteria to outer space, and then we're going to look at the gene expression differences.
We really love outer space, and we want to protect our astronauts. We're also really passionate about the study of infectious disease, so we thought that this project would be the perfect intersection of all of our interests.
We hope to learn how bacteria respond to antibiotics in space and how we can better treat infections that may occur during spaceflight. We're really excited to see the results of our experiment. Hail to Pitt Pharmacy!
I'm Dylan McKenzie.
I'm Curtis Lynn.
And I'm Anthony Ebner. And we are the fun guys from the Student Spaceflight Experiment Program in Laverne, California. Our experiment is studying Clamulina phyllotypes growth in microgravity. This is the common enoki mushroom. The two main objectives of this experiment will be to see how the observed growth patterns of the mushroom can help in the propagation and cultivation of mushrooms in space, and to prove or disprove two theories on how mushrooms can detect gravity. These two theories are David Moore's theory of mushroom gravimorphogenesis, which states that the apex of the stem is a part of the mushroom which detects gravity. The other theory is Jan Monzer's theory of mushroom gravitropism, which states that the nucleus of the mushroom cells acts as an otolith which detects gravity. These theories will be tested by growing the mushrooms both aboard the International Space Station and on the ground. Upon the end of the experiment, the growth patterns of the mushrooms will be examined and studied to see which of these theories are true and what we can learn about growing mushrooms in space.
We came up with this idea when I watched a video on PBS Digital Studios discussing how plants sense gravity. And I was wondering, how do fungi sense gravity as they are in a different kingdom, so they might have a different method to do so?
This experiment will help us gain insight on how exactly fungi is able to detect gravity compared to plants. Also, this experiment will help us understand whether Moore or Monzer's theory of
fungi gravitropism will hold up. Thank you for listening to us, the fun guys, in our experiment for the SSEP program on familial volatiles in microgravity.
I'm Madison Morgan.
I'm Abigail Craig.
We are from the Student Spaceflight Experiment Program from Ocean City, New Jersey. We chose to investigate the effect of microgravity on the hatch rate and development of Artemia selena.
We're sending brine shrimp or Artemia selena to the International Space Station to test how far they grow in their life cycle, and then to test the sex ratios between males to females.
We chose to use brine shrimp for this investigation because it has been used in the past and has been proven to be an efficient source for experiments, and through this research is also very inexpensive. Knowing that the brine shrimp, that they're very small in size and don't take up a lot of space, we believe they could be a good source of energy to the astronauts and an easy way to clean the shuttle.
We also know that brine shrimp, or Artemia selena, cysts, can be preserved for long periods of time, which would help when we eventually colonize Mars. That is why we chose to study the effect of microgravity on the hatch rate and development of Artemia selena.
Students at a Parksville high school are taking the concept of higher learning to a whole new level. They've been designing an experiment that will soon be aboard the International Space Station.
We are Sprouts in Space from the Student Spaceflight Experiment Program in School District 69, Qualcomm in Parksville, British Columbia.
So the title of our experiment is Investigating the Growth Patterns of Alfalfa Sprouts in Microgravity, a Potential Nourishment for Future Manned Spaceflight. The experiment will be performed in a Type 3 FME mini-lab. We will compare germination rate, root length, cross-sectional area, volume, and collective mass between the ground control and the experiment itself. This experiment will attempt to demonstrate how alfalfa sprouts react to the conditions in space.
For this experiment, we want something lightweight and compact with minimal growth requirements and a high cost efficiency. With lots of research and investigation, we found alfalfa sprouts to contain all of these properties as well as be a viable source of nutrition.
As all of us are fascinated by space exploration, we designed an experiment aimed to benefit astronauts on long-term journeys. We think that it's important to find plants that grow effectively in microgravity so that our pace of exploration can increase.
This experiment could provide the scientific community with data that could allow further investigation into how alfalfa seeds germinate in microgravity to eventually confirm its potential in future long-term space flights.
We are the John F. Kennedy Elementary School team from the Student Spaceflight Experiment Program in Riverside, California.
Our hypothesis is that crystals grown in microgravity will be smaller and have less flaws than those grown on Earth. This is because they will not sink to the bottom of the tube and they will grow extra slowly. We wanted to do this project because no one had done it before. Us kids could do it, and if it worked, it could make better tests for testing anemia, and it could make better fiber optic cables.